All right. So we're going to get going. We said that for most traits, there's a dominant trait and there's a recessive trait, right? We talked about hitchhiker's thumbs and we talked about tall pea plants and short pea plants. And that is sometimes true, but sometimes they're not. So we're going to talk about it. And we're going to say that some traits do not exhibit a fully dominant trait. In some cases, a heterozygous individual will exhibit a blend of those two traits. Sort of Lamarckian in nature. Yeah. Am I in the way? How's that? That's probably worse for you. How's this? Oh, good. I'm glad. So we call this partial dominance. Sometimes we also call it incomplete dominance. Those are synonyms. Physical traits are often partially dominant. In other words, if you come from a blended family and one of your parents has dark skin tones and another one of your parents has light skin tones, often the offspring will have skin tones kind of somewhere in the middle. Hands up if that describes you. Skin tones are kind of in the middle of your parents. Is there anyone here who has, parents with two different skin tones who are distinctly more like one than the other? Yeah, in your case, you're distinctly more like your dad. Yeah, exactly. And what's your dad's background? They're both Indian, but he has darker skin tone than she. Interesting. Yeah. So in this case, we get something called partial dominance. And this is actually good because we'll be able to describe both of those situations, both a case where your parents have two different skin tones and yet you seem to end up just having one of theirs, and then another case where they have two different skin tones and you seem like a blend of the two. Both of those make genetic sense, and we'll look at how. But we're actually going to use an example first of curly hair in humans, which is actually largely, though not completely, controlled by a single gene. So curly versus straight hair in humans. All right. And by convention, we often won't use, so if curly was dominant and straight recessive, what would we use for the alleles? We would probably call the curly allele, if it was dominant, if it were dominant. We'd call it big C, and what would we call straight hair by that convention? Little c. But in this case, because one isn't dominant over the other, we say curly is partially dominant. We have a second convention we use sometimes where we'll say HC is the curly allele, and HS is the straight allele. All right, so if those are the alleles, what are the possible genotypes? And what are the possible phenotypes? But first we'll get to genotypes. Well, how many copies of each gene do we have in our cells? 2, because we are, what's the word to describe organisms that have 2 copies of each gene? Diploid, because we're diploid. So what are the possible combinations you could have? Come on now, it's simple, yeah. Yeah. Yeah, very good. You could have 2 copies of the curly allele, so we'd say HCHC. And what's that person going to look like, obviously? What's their phenotype going to be? They're going to be curly-haired, right? And the other possible alleles would be what? Yep. Max?
HS and HS.
Very good. In that case, we would obviously say that they have what kind of hair? Straight hair. And we've dealt with the 2 cases where the 2 alleles are the same. What do we call both these? Both of these are examples of being homozygous. Very good. But of course, what's the other possibility? Being heterozygous, which in this case would be what? HCHS. Does it make any difference if we write HCHS or HSHC? No. Now, in a case where one was dominant, we would end up either being curly-haired or straight-haired, but in this case, we actually have what's called this blended dominance, or this partial dominance. And in the case of partial dominance, we say that curly is partially dominant over straight. It changes it. Yeah.
Do you have any meter sticks that you never use again?
No. Sorry.
You don't need to use them to scare kids.
I mean, you can borrow a meter stick if you want.
No, we already have one. We're trying to find a permanent one.
Are you really wasting my time with this? Go away. I love you. Go away. Okay. Was that too harsh? That felt harsh. I'm sorry.
No,
You literally said I love you as well.
Yeah, you said I love you.
It's so cute.
Okay, anyway. But I also say it was wasting my time, and I said, go away. That felt harsher.
And then you said I love you.
And he's a nice boy. Okay, anyway, we'll focus. Anyway, so what does somebody who has 1 copy of each look like if one isn't fully dominant over the other? And the answer is, yeah, kind of wavy-haired. That's a V. You wouldn't know it to look at it. Looks more like a check mark. My 6-year-old Charlie was making fun of my letter A's the other day. And he said, his teacher doesn't draw them like that, so I must be a bad teacher. That hurt my feelings. And I said, Charlie, I am a bad teacher, but that's not why. Anyway. Then he was really upset. He was really worried that I was a bad teacher. It's odd when he says it, it's fine. And then when I say it, it's all upsetting. Charlie, if you're watching this, I love you. Now stop calling me a bad teacher. Presumably, if he's watching this, it will be like 11 years from now when he's in grade 11 biology. So, how you doing? Anyway, okay, so that's a case there, and we can look at that. And traits like this exist. There's lots of them. Like we said, skin tone, which skin tone is actually controlled by a lot of genes, which is why we don't use as an example here. There are other things that affect your hair straightness, but there is a single gene that really does have 2 alleles rather, one of which sort of is for very straight, one of which is for curly, and if you have one of each, you're wavy. There are other genes that will still impact hair straightness in other minor ways, but there's one kind of major gene that largely controls it, which is interesting. And that's why we use that as an example. Though you'll find by the time you get to human systems, we are controlled by so many genes that very few traits are like fully controlled by one thing. Does that make sense? And then diet affects your hair texture, and how you wash it and condition it affects your hair texture. And if, maybe hypothetically speaking, you spent all the late 90s and early 2000s with a middle part, then you're never going to get rid of that thing. These are the things. Anyway, so that's one possibility. There's also a second possibility, which is a situation that we call codominance. And we're going to look at this as our second example. So I should have put a big old 1 by partial dominance, but that's okay. I'm going to put a big old 2 in front of codominance, and we'll go back and change it later. And you know what? I'm going to make it a Roman numeral. Try and stop me. That was like the exact opposite of trying to stop me. It was just whatever, in front of partial dominance. Stop living in the past, man. It's time to move into the future. God. Okay. In codominance, there's more than one dominant allele. It's not that no allele is dominant. It's that there's more than one dominant allele. And we'll make a note, there may also be some recessive alleles. There can be more than two versions of a gene. And in this case, heterozygotes will exhibit all dominant traits. Now, that doesn't always make sense, and which is why not every gene can be codominant. For example, you couldn't have being tall and being short both being dominant traits. Because if you had one copy of one and one copy of the other, you can't be both tall and short at the same time. Does that make sense? That doesn't work. Wavy-haired and curly-haired, I mean, I guess maybe like the left half of your head could have straight hair and the right half would have curly, but by and large, you can't have both at the same time. Do you know what I mean? But there are some traits where, of course, you can have both at the same time, right? And I'm going to look at a classic example of this, which is blood typing. Okay, so exempla gratia, human blood typing, or at least half of it. In fact, we're going to look at human blood typing altogether in just a minute. But human, what we call ABO blood typing. Have you heard of that before? We'll look at this further in our animal systems unit. In fact, we'll do a lab where we use simulated blood and test it to see if it's type A, type B, or type O, and show how that's actually done in the lab. But of course, we won't do it with real blood because that's dangerous. So we'll use synthetic blood. But human ABO blood type, I should say type, not typing, sorry. Type is whatever. Nobody will know if you don't change it. Well, we'll talk about that. It's controlled by one gene with 3 alleles. One gene with 3 alleles. And to the best of my knowledge, maybe in some of the homework questions I gave you, have you seen a question where there's 3 alleles in one gene yet? I don't think so. But I mean, you can have a gene with 50 alleles. In fact, most genes have lots and lots and lots of alleles, but we group all the ones that are very only minor changes all as the same. Because, of course, a mutation causes an allele, right? Keep in mind that on some chromosome, here's a chromosome, right? Here's a chromosome. We could look at a specific section of it. I don't know, we could zoom in here. And if we zoomed in here, we'd see, you know, there's some DNA, and DNA, of course, looks like this double helix, right? And then, if we got down to it, we could sort of look at that, right? And we know that the DNA itself actually looks like A, G, C, C, G, T, T, A. I don't know. By the way, what would the complementary strand here be? Oh, whoops. Cool. All right. So there could actually be 3 different versions of this, right? Like 3 different lines of code just remember that when we talk about a different allele, that's actually what we're talking about, right? Like that we're actually looking at the DNA and it's written differently. It's a different line of code. So this might be one version of the allele, and then another one, this section here is replaced by something different. And maybe in another one, this section's replaced by something different. Does that make sense? And it causes a change in trait. And as it happens, we have 3 alleles. The first allele is the A allele, and this codes for type A blood. We'll learn more about that next unit. For now, it's enough for you to know that it codes for type A blood. Does anyone know what it means by blood type? Yeah. I usually talk about when I teach grade 10 science, so I don't always, but usually I do.
Blood that is like...
It has type. Got it. Agreed. Yes.
Is it like protein signifiers?
Sorry?
Is it like signifiers in the blood?
Yeah. Oh, very good. Yeah, that's exactly right. On the red blood cells themselves, so this is a red blood cell, and again, we'll learn more about this next unit. If you ever look at red blood cells under a microscope, they have this kind of donut-like appearance to them. There's not a hole in the middle, but they get thinner. They have little flags on them, little markers, and they can have little flags on them that have an A on them. And if they did, that would be type A. And they're literally used, cells have all kinds of markers like this to signify what type of cell they are. So the A antigen codes for those type A blood markers. The B antigen codes for, yeah.
Do the blood cells, they actually just have things that say A, B, and O?
I'm not answering that question. Okay. Are you asking, are there little plastic signs on them with the letter A written on them?
I mean,
I'm not answering that question. Well, that's a silly thing to get from that. What they have is long chains of carbohydrates, glycolipids that come off of them that we call the A antigen. And then there's another version where instead of having those antigens, they have what we call the B antigen. And the B antigens, I don't know, they look different. Maybe they look like, I don't know, this. Look at me, I'm a B antigen. Which again, it would be really surprising if they used the Roman alphabet that wasn't designed until, about 2,000 years ago, all over the cell. So no, it's not actually written as a little B on there. I got to be honest, and listen, speaking of people I love, I love you, right? I cannot believe I had to answer that question today. My faith in our education system is shook. I'm going to go home and think about this, and I'm going to ponder my life choices. I'm going to ask, is this how I want to be spending my life? And I got to be honest with you, I don't know what the answer to that will be. Anyway, but that moment changed my life. I just want you to know that. Okay. No. Let's carry on. And then what did we call the third allele? Well, A, B, and O. Why did we switch to the letter O? What does it code for? Yeah, but what's that mean? The interesting thing is actually this. It codes for nothing. In other words, it's just kind of a broken gene. And we call that type O, but the reason they don't use type like C is because it's not like it has an O antigen. O blood is actually interesting because it just doesn't have any markers at all. I mean, it does have markers, but it doesn't have A markers or B markers. There's markers that all your cells have on them. Every cell in your body has some kind of marker on it saying, hey, I'm this kind of cell, notice me, pay attention to me. Like name tags. All your cells have little name tags on them. But again, to be clear, they're not really name tags because that would be silly. Yes.
So, when it comes to O positive and O negative.
We'll get to the positive negative in a minute. Just hold up. It's a good question. Unlike other questions which were bad questions, that was a good question. It's just not a good question yet. Unlike yours, which was never a good question. Do you see the difference? Good question too soon. Bad question. bad. Questions so bad it's making me ask questions like, why? Why, Joe? Why? Anyway, I'm glad I'm recording this. This is good. I plead guilty, Your Honor. Charlie was right. I am a bad teacher. Charlie got really mad at me one day, and he wanted to watch TV, and I said no. And he said, you need to let me watch TV. And I said, I don't need to do anything. And he said, you're a bad dad. And everyone's gonna know that, and they're gonna make a big circle around you, and they're all gonna point and tell you you're a bad father. And I thought, wow, this kid knows how to go for the jugular. Anyway, I didn't let him watch TV, but it hurt my feelings. Yeah. But now remember, by the way, as it happens, this gene lives on chromosome number 9, if you're wondering. The ABO blood gene is a gene on chromosome number 9. This gene codes for markers on the outside of red blood cells. These are called antigens. You don't need to know that, but so now, again, keep in mind that the A cells, or the A antigen, the A allele rather, codes for A markers, A antigens. The B allele codes for B markers, but the O allele codes for nothing. Now, the thing of it is this. These are both dominant. Because if your cells have this gene, then the red blood cells will make A markers. This is dominant. If your cells have this gene, they will make the B marker. O, however, just doesn't do anything, which means if you have the O allele, it won't do anything. Now, if you have 2 of the O alleles, then will you have any cell markers? No, but if you only have 1 of them, it would not affect you in any way, shape, or form. So we say that trait is recessive. You have to have 2 copies of it for it to matter. Well, you should see pretty quickly that we end up with a bunch of possible genotypes, right? All right. So what are the possible genotypes here? There's actually a lot. By virtue of having 2 choices with 3 choices to make each time, how many combinations is that, you mathletes?
Wait, what?
Well, you get to make a choice once, and it's a choice between A, B, or O, so 3 options. Then for each of those, you make a second option of A, B, or O. How many possible choices is that? Is it 6? Did you say, do you want a combination or a permutation? Did somebody ask? I'm just wondering, is somebody in data management right now? Oh, I thought I heard the data management term. I was like, ooh, fancy. But you don't need to be that fancy. I mean, listen, don't draw this. Put your pens down. Your first gene, it could either be A, or it could be B, or it could be O. That's a terrible drawing, but whatever, we'll go with it. It could be A, it could be B, or it could be O, right? Now, once your first one was A, what possible choices do you have then? The next one could be A, in which case you would have AA, right? The next one would be B, in which case you would have AB. Or the next one after that? O, in which case you would have AO. AO, okay, there we go. If your first one is B, what choice is that? Well, next one you could choose A, in which case you would be AB again, right? BA and AB are the same thing. So we'll make a note there. That's our first duplicate. What if your second choice was B? What could you be then? BB, all right? And your next option? O, in which case you would have... Oh, you'd have BO. Unfortunate. Unfortunately, those are my wife's initials. It's problematic. Britt Orlando. BO. Her middle initial name is Elizabeth, so her initials, when spelled out, say the word BO. She can't win. Anyway, yeah. Patriarchy, am I right? Okay, and then what's the option here? You could get O, and then you could get A, in which case you would have Hey-oh, there's another duplicate, right? And you could have O and then you could have B, in which case you'd be BO again. Oh, there's another duplicate. Another highlighter. Oh, I haven't used that one yet. And finally, you could have O and then get O again, in which case you would be Oh, there we go. That was good. You know what? I don't hate that. So you do not have to draw this, but I'm actually going to, you know what? I thought this was a sidebar, and I'm just going to throw it in the note because you know what? If somebody's looking at this, they might find that helpful. Well, that's unfortunate. Oh, golly. I'm very folksy. Third thing people notice about me. But I'm tough, so watch it. They notice I'm tough again. I'm just that tough. Okay, so the possible genotypes then are AA, AB, AO, BB, BO, or OO, right? Okay, so the possible genotypes then would be AA, in which case you'd be type A. And if we drew your red blood cell, schematically, of course, because you know that your red blood cell is also not made of ink. Fun fact. You might be shocked to learn. And it would have our little A antigens on here, right? Those are my little A antigens. Aren't they pretty? Very pretty, right? Okay. What else could you be? Well, we said you could be AB, right? If you're AB, then what blood type are you? Well, we said these are both dominant. So in this case, you would be type AB. AB. There we go. Okay, there we go. And this would have those A antigens, which I believe I drew as blue squares. But then they also would have little, I think I did those as green circles. Am I remembering that right? That would be type AB. In other words, your cells just have both markers. Can a cell have two markers on it? Yeah, sure. Okay, but our third genotype was AO. What blood type is this person? Yeah, they're just type A. There's no such thing as type AO because O is recessive. So they are just also type A. Okay, what was our next possible combination? We had BB, right? And obviously, what type blood are they? Type B, right? The other possibility was BO, and they are actually also type B. I'm just going to draw those next to each other. Save some time. And what are they going to look like? What are they going to look like? Yeah, they've got a little green B antigens on them. This is nice because when I teach this lesson next unit, it's going to go so fast because you're just going to already know it. And then what's the last possibility? We've done AA, AB, AO, BB, BO. And finally we have OO, and that is just what we call type O. So of the 9 possible ways they can come together, one is type A, heterozygous, but actually 1, 2, 3 of them are type O. 1, 2, 3 of them are type B. 2 of them are type A, B, and only 1 of them is type O. So you get this very specific ratio by virtue of there being 3 alleles, with 2 of them being dominant and 1 of them being recessive. So not that we asked for the frequencies, but let's look at the frequencies. Oh, out we go. All right, team, let's head on out. Doing a nice orderly fashion for the doors. Only a drill. They said that. Get away from the building, treat it like it's a real fire.
Oh yeah, I just couldn't stop it. Interrupted our list.
I said, wow, Angela, that's low.
He's 14.
Don't let him get to you.
All right. So as we were saying, these are our blood types. So now, if we wanted to, we could look at these, and do a hybrid cross, right? If we want to, right? We could say, I don't know, let's make one up. Let's say we have a type O father is crossed with, which feels sort of euphemistic, is crossed with a type A woman whose 23 siblings are all also type A. Yeah, she has 23 siblings, so she's 24 of them. A lot of kids. I'm just going to say, what would the F1 generation look like? No, we're not making 23 kids. That's to give you a hint. She's type A, but what is probably true if she is type A and all 23 of her siblings are type A? What type of type A is she? Because, of course, how could she be type A? She could be type A this way, but she could also be type A this way. So, having said that, if all 23 of her siblings, and let's say, and cousins, just to really sell it. So what is her genotype likely? AA, right? That's what I'm trying to drive at here. I just want to tell you. We gathered some evidence. Okay, how do we figure that out? Well, by convention, we usually put the mom up on the top of these things. So mom is here, and we're saying she is type AA. We think mom is heterozygous, is a good guess. Now, is it possible that she's not? Sure. Maybe her mom is heterozygous dominant. Yeah. Hi. Yes. Oh, thank you. Oh, bless him. And he gave me 4 of them back, despite me printing 21. That's very helpful. Thank you, Alfred. Oh, did I say hetero? What do I mean?
Yeah, I was like, wait.
That's because I was thinking ahead of my question, which is it possible that she could be heterozygous and still have all siblings who do? Of course, right? But we're going to assume. We're assuming. All right, without doing a genetic analysis. So now, if mom is producing egg cells, if mom produces ova, by what process? What process produces, well, sure, but we'll talk about, she's not ovulating yet. When she actually made the ova, what's that cellular process? Meiosis. Very good. Right? Because in meiosis, see, I tried to cheat and just turn the S into an O, and that was lazy, and I paid for it. There we go. Okay. When she produces that, she is diploid, right? But are her ova diploid? Are egg cells diploid? Do they have 2 copies? No, they don't. They are, what's the other word? Haploid is correct. So, oh my God, that's nightmarish, that angle at the bottom. I don't know why it doesn't give a lot of leeway to defaulting to just straight lines. Anyway, there we go. So when mom produces eggs via meiosis, those eggs will be haploid. Though in this case, she's either going to pass on a big A or she's going to pass on a big A. Okay, not very exciting, right? But even still, that would be the big A from her mom, and that would be the big A from her dad. Okay, what's the dad in this case? Dad is double O. Not very exciting. But we don't have to assume here, dad must be double O. Right? And we'll just say homozygous. And the reason you have to be homozygous to exhibit the recessive trait is because it's the recessive trait. So now, if he produces spermatozoa, by meiosis, his sperm cells will either have big O or they'll have big O. Okay, not very exciting, but there we go. Yeah.
If O is recessive, wouldn't they know?
You know what? That's just how they do it with blood typing. It's kind of, you're right, it breaks convention. I don't know. You're right, it breaks convention. It's just how it's done. Yeah, I think the fact... Yeah, I think it just... The truth is, yeah, that this is, I don't know, kind of a special case. And so what genotype do we get here? AO. What about here? What about here? What about here? Interesting. So, if your parents are both homozygous for different traits, all of you and your siblings will be heterozygous. Just has to be, right? Does that make sense? Yeah. So in this case, 100% of the F1 generation is AO, right? And what's the phenotype associated with that? 100% of them will be type A, which actually means that all of mom's siblings being type A actually isn't evidence that she's type A, though if we factor in all her cousins and stuff too, we can be pretty confident. She has cousins, yeah. The names are Larry and Gary and Barry, Harry, and Jim. Carrie. Carly. Okay. Anyway, now, do we have time? We got time. I have faith. We're going to do part 3, and we're going to call this dihybrid crosses. Does anyone know their blood type? I like mine because my blood type is an instruction. It's B positive. So I think about that from time to time. I'm like, my cells are telling me to be positive, which makes me feel like a traitor when I'm so negative all the time. But I can't help it. Because a kid asked me today if there's a little letter A written on the side of my blood cells. I'm going to think about that tonight. Anyway. All right. So we're going to build up to this by saying blood type actually has a second gene, which you know, because you didn't just tell me that you're type O, you told me that you're type O positive. And I didn't just tell you that I'm type B, I told you that I'm type B positive. Yeah. So blood type is actually controlled by a second gene as well. A second gene, this one with 2 alleles. It codes for another marker called the Rh factor. But this one is a whole different gene. It's called the Rh factor because it was discovered in whatever these monkeys are. I never know how to say that word. Anyway, we just call it the Rh factor. Shh, quiet. So, there's 2 alleles. And we write this very creatively as Rh positive, and this codes for, this is dominant, and it codes for the Rh allele, or the Rh antigen, rather. Actually, I'm not even going to write that. So I'm just going to say, no. I'm just going to say, I'm going to say positive blood type, because I'm going to draw my picture. My pictures are so good, right? You don't want to be deprived of that. No. Right? Yeah, no, you don't want to be deprived. And scientists were amazed to discover when they looked under the microscopes that what this actually looks like is a little RH just on the end of a stick on each cell. It's the craziest thing, and that's why Karthik's question was so surprising to me, because I thought, how did he know? But it wasn't for ABO blood typing. That's what RH blood type looks like. But you can see, like, you got it really close. Anyway, yeah, it really amazed scientists. Like, who knew that our alphabet evolved in our blood cells? Right.
You're not being serious, right?
No, I'm not being serious.
Sorry, I just have to make sure.
Kartik, I love you dearly. Wow.
I'm a little toned down.
And recessive is just another one that's broken. It codes for nothing. And because it codes for nothing, we say that it is negative. In other words, if you have the Rh negative antigen, you do not have the Rh antigens on your blood cells. But mind you, you could still have what markers on there? A or B or A and B. Does that make sense? They're controlled by two totally different genes, which is why you have to say you're type A or type B or type AB or type O, and then you also have to say that you're positive or you're negative. They're controlled by two different genes. Could we track that? And I think we have enough time, so I think we can. So now, let's track two traits.
No, that's impossible.
No, there's no going back, only going forward. How's that? Let's track two genes at once, and we call this a dihybrid cross. Shh, quiet. Quiet. Thank you. In a dihybrid cross, it doesn't just mean that we're crossing two traits. Of course, when two animals mate, when they reproduce, they cross all of their traits. But it means that we're tracking two of them. Does that make sense? We're going to look at two traits at once. Is that going to make it more difficult? Well, of course it is. But that's okay. Are we afraid of that?
Yes.
No, we're not. I'll make this one easy, which is we'll say mom has this genotype. Ready? Now, how many genes are we talking? We're going to talk the ABO gene, and we're going to talk the RH gene, right? But how many copies of each? No. She's a human. How many copies of each gene do humans have? So in this case, she's going to have A and B here, and she's going to have an RH positive allele, and she's going to have an RH negative allele. Does that make sense? What is her blood type? AB positive, right? A and B are codominant, so she's type AB, and positive is dominant over negative, so she's type AB positive. If we looked at her blood cells, she has all of the antigens a person's blood cells can have. So her little cell sign pasters are working overtime because they've got the little B antigens, right, which I showed as green dots. There they are. And they've got the little A antigens, which I showed as blue squares. Right? And then she's also got the RH factor all over there like this, right? She's got busy blood cells. They're covered in antigens. Okay? Let's consider Dad. Shh. Dad has, now we're going to make him A negative, but he's going to be A by virtue of being AO, and he's going to be RH negative, RH negative. So Dad is A negative. Okay. So what do his blood cells look like? Yeah, which I coded, those are my blue ones, right? Not that it matters, because of course these are just schematic, but at least to be consistent. So we'll show Dad's blood cells have only the A antigen on them. Okay. So this is interesting because we have an example of we're tracking one gene that has 3 possible alleles, another one that has 2 possible, but that's okay. We can make a Punnett square, but let me ask you, before we draw the square, let's see what mom's ova could look like. So we know that mom's genotype is AB, RH plus, RH minus, right? And I'm actually going to color these to make seeing the combinations easier. I'm going to say, you don't have to do this, but I'm going to call her A red. There's nothing I guess it does not matter. I'm going to call her A red, and I'm going to call her B orange works, okay? And I'm going to call her RH positive blue, doesn't matter. And I'm going to call her RH negative this purple. It doesn't matter, just to help us kind of see where they're coming from. When she goes to make ova Through what process? Yeah, via meiosis. Mom will produce ova. How many ways could these come together? Well, a single ovum will either have her A allele, right? Or it will have her B allele. But it will also either have her Rh plus, or it will have her Rh minus. But listen, does the A have to be with the Rh plus? What else could the A be with? Don't be talking over me. Thank you. The A could also just as easily be with the Rh minus. And does her B have to be with the Rh minus? No, it could just as easily be with the... Quiet. I just said be quiet. Was it confusing the first time? No? Then be quiet. All right, unless you're answering my question, just be quiet. This is confusing stuff, so we don't need second conversations going so nobody can focus. All right. So these are the possible combinations. In other words, how many ways, if we're tracking 2 genes, can mom make an egg cell? 4. She could have allele 1 of the first one with allele 1 of the second one, or 1 and 2, or 2 and 1, or 2 and 2. So if we're tracking 2 traits, for a dihybrid cross, there's 4 possible gametes. Well, that means if mom gives us 4 gametes, how many gametes is dad going to give us? 4. What's a 4 by 4 grid? How big? 16. So this is going to be a fairly complicated thing, but that's okay. We got it. We're going to say, okay. 1. That was terrible. Oh my gosh. 1. 2. 3. And 4. And
Ms. Ferguson, please contact the office.
okay, there we are. That's my almost perfect square. Okay, now what about dad? What was his blood type? He was type O. All right. And what was his? He was A negative, which means he has Rh negative and Rh negative, right? Okay, so either the A allele will end up paired up with the Rh negative allele, in which case his first gamete would be A Rh negative. Or his A will pair up with Rh negative. So that one is, not very exciting. O can pair up with Rh negative, or O can pair up with same thing. So this one, at least, we only actually have to work out 2 rows, right? And then the other 2 rows will just be doubles of it. Does that make sense? Okay, so let's work out this top one. What will we get here? We'll get A with A, giving us AA, and we'll get Rh positive with Rh negative. What blood type will this person be? A positive. Good. Okay, what do we get here? A will pair up with... A pairs up with B. And Rh negative will pair up with... Very good. What blood type is this person? AB negative. Good. Okay, what about here? A pairs up with A, and Rh negative pairs up with Rh negative, and this will be A negative. And what about here? We'll get A with B, and we get the Rh positive and the Rh negative. What type will this be? AB positive. Okay, this will be the same as above, right? Should I fill it in? I guess I should fill it in, right? Why be negative? Why be lazy? No, you know what? I am just good because I'll show you. It actually is going to save us a lot of time because then we can just count the above one twice when we go to our counting, right? Why be cute? What will this be? Yeah, by convention, we still put one of the dominant ones first. So we'll go AO, and then we'll say RH positive and RH negative. What blood type are they? A positive. And here? BO and RH negative, RH negative. This person will be B negative. Here we go. A, O, RH negative, RH negative. This person will be A negative. Here, BO, H positive, RH negative. This person will be B positive. And these will just be the same. Now, is that a bit of a pain? Yeah, I guess. But it's not any more difficult than a monohybrid cross. The main thing to keep in mind, though, is this. Listen, look at me. Mom is a person. And a person has to have how many of each gene? 2. So she has 2 blood type, ABO type genes, and she has 2 RH factor genes. Dad has 2 blood type ABO genes and 2 RH genes, right? On the other hand, is an egg cell a person? No, it's just an egg cell. Is an egg cell, bless you. Does an egg cell have 2 copies of every gene? No. So at the top and the side of any Punnett square, whether it's a monohybrid square or a dihybrid square, there should only be 1 copy of each. By the way, we're going to have a quiz again on Friday of this week. But the last quiz, some people had like bonkers stuff going on in Punnett square. I can only assume that means you didn't do your homework. Because the homework had the answers in the back. And like, you didn't come to me and ask for help. But like, you're in a grade 11 university level course, right? So if I give you homework and say, make sure you can do this, the expectation is you can do this. Now you're gonna get a little assignment package so we get some practice in before the test, but I wouldn't want my first practice to be on the assignment that's being marked. I would want my first practice to be on homework, right? Practice. And then a quiz, because quizzes aren't worth much, and then I want to do the assignment package when I feel like I'm good at it, right? So make sure you're doing the homework. So each of these is a possible egg cell. How did we get the egg cells from her blood type? Well, one of these has to pair up with one of those, and there's 4 ways to do that. You could actually build a little Punnett square to see how these come together if you wanted to. You could do A and B at the top and RH positive and RH negative on the side and see the 4 kinds of egg cells she can make. If you struggle with this visually, use a Punnett square to figure out her egg cells, and then from there, use another Punnett square to figure out the offspring. Does that make sense? Yeah. Yeah, of course. I'm pro-punching. Everyone knows that. So now, let's talk about the F1 generation. The F1 generation, what are the genotypes we have here? Well, we have AA plus minus. And how many times does that occur? That's twice, right? There and there. So we get AA plus minus. And that is 2 out of, how big is this grid? 16, which is an eighth, which is 12.5%. Half of 25%, right? Cool. We have AB minus minus. How many times do we see that? Well, twice, right? So we have AB, RH minus, and that's going to be 2 out of 16, which is 12.5%, right? And then we have AA minus minus. How many times does that occur? Twice. Boy, this is going to get tiresome, eh? And yet, we're not descended of cowards. We're not afraid. Speak for yourself. I'm tough. First thing people notice about me. They're like, you speak for yourself, Orlando. I'm a coward. And this is 2 of 16, and that's 12.5%. Is everyone comfortable with the fact that I didn't bother filling in this row because it's just the same as the one above it? Anyone desperate for me to fill that row in? I won't do it. I refuse. Just so you know, if you're wondering where that request ends, it's with me saying, we're not cowards. You don't need me to do that. I'm going to stop highlighting because can we see that in this table, everything appears exactly twice? We have AO Rh plus, Rh minus. And that occurs 2 out of 16 times, which is 12.5%. Then we have BO, Rh minus, and that occurs 2 out of 16 times, which is 12.5%. And then finally, we have two more, not finally, penultimately. Do you know the word penultimate? What does penultimate mean? Very good, second to last. Don't actually, that was rude. Actually, that's so, listen, Kartik does not remotely lack for cleverness or intelligence or knowledge. He just sometimes speaks a little faster than he thinks, which, for the record, is a thing I relate to. So, Kartik, I may give you a hard time, but it's just me giving you a hard time. Nothing but respect. Actually, how dare you? And then the last one would be BO, and that one's RH plus, RH minus. And that is 2 out of 16 times, which is 12.5%. Boy, my percentage signs are looking rough today. Okay, those are the phenotypes. We could also, of course, track the genotypes. That's a little bit better. How many genotypes do I have? I've got A positive. How many A positives are there on here? 1, 2, 3, 4. So 4 out of 16. What percentage is that? Very good. All right, I have, what else? I have a negative, and that is 1, 2, 3, 4. So 4 of 16, which is 25%. And then I have AB positive, of which there are 2, AB negative, of which there are 2, B positive 2, B negative 2. So B positive, there's only 2 of 16. B negative, there's 2 of 16. And then what was the other? AB positive, there was 2 of 16. And AB negative, there's 2 of 16. And these are all 12.5%, right? Okay. So first things first, hopefully you learn from this that there's not a lot of detective work to be done about your blood type. So unless you have 200 siblings to start getting percentages from, don't go home and be like, there's some deep family secret because my blood type's different than my sister. You're good. Lots of possible blood type combinations. But 2, tracking 2 traits, a dihybrid cross. Tricky? Okay, the TLDR of today. We learned that some traits, the heterozygous gives us a blend of the 2 traits. We call that partial dominance or incomplete dominance. Sometimes, when you have a heterozygous, you just get both traits. We call that codominance. Then, we looked at ABO blood typing, and then we decided to add the RH factor and to look at an example of tracking 2 traits at once. And we call tracking 2 traits at once a dihybrid cross. We still use a Punnett square, but now how big is our Punnett square? Well, it's not 16 by 16. That would be a nightmare. It is not 8 by 8. That would still be a nightmare. It's 4 by 4. Can you imagine if you had to track all 138,000 coding genes on the genome?
I would cry.
Well, and the answer is we can't do that. The math is too complicated. There's some practice worksheets here. Interestingly, there are 4 of them. I will print more right now on my printer. But grab one. I want you to do this dihybrid cross practice, all right? We want to get good at this. If you come to me and say, hey, I did my homework, here's a question I'm having trouble with, I'll help you. But if listen, I'm going to go to my class or not. Barry, thank you. What's your language? I'm the one who cusses in this room. All right, we'll do the rest later. Have a great day. So did you ask me? Barry, back in Barry. Oh, I'd love that. Yeah, you bet. Thank you. Okay, I'll print for you right now. Give me just a second. I'll print you some of this stuff. Yeah, I've got a few 3D printers.